Chapter 2, Biological Classification. Let's see important points in this chapter. Aristotle. Aristotle is the earliest for scientific classification. What he did, he divided into plants and animals. Plants includes trees, shrubs and herbs. Herbs means smaller plants, shrubs medium heighted, and trees are very long. So whereas animals, he divided into two. One is red blood cells, another is non-red blood cells. And three kingdom classification is given by Ernest Haeckel, and two kingdom by Linnaeus, and four kingdom by Copeland, five kingdom by Whittaker, six kingdom by Carl Woese. Two kingdom classification includes, we all know, Plantae and Animalia. What are the drawbacks? Drawbacks are prokaryotes, they included prokaryotes, bacteria and blue-green algae, and eukaryotes brought together. Prokaryotes, example bacteria and blue-green algae, and eukaryotes are combined in this group. Okay, next one is unicellular and multicellular organisms. Unicellular like Chlamydomonas and multicellular like Spirogyra are both joined together into a same group. And photosynthetic and heterotrophic, there is no separation between photosynthetic and heterotrophic, fungus and plants. So these three are the drawbacks of two kingdom classification. And Carl Woese, who gave six kingdom classification, actually uses 16S RNA, 16S ribosomal RNA for six kingdom classification. Carl Woese divided the Monera into two kingdoms, Archaebacteria and Eubacteria. That means in Whittaker classification, he divided the Monera into two groups. So it becomes six kingdoms. Next, Carl Woese studied six kingdoms and observed that naturally clustered into three main categories, that are bacteria, archaea and eukarya. He divided the organisms into six groups, even though after division he gave three main categories to which these six kingdoms belongs to. Example, bacteria, archaea and eukarya. And archaea and eukarya are more closely related. So what is the criteria for five kingdom classification? The most famous classification was five kingdom classification. Based on what? Based on what are the criteria for this five kingdom classification was cell structure, thallus organization, mode of nutrition, reproduction, phylogenetic classification. What is meant by phylogenetic classification? It means it is a evolutionary relationship between the organisms. So next, during unfavorable conditions, bacteria produces endospores and fungi produces exospores. It's very important. Bacteria produces endospores and fungi produces exospores. So in this chapter they will give five kingdoms: Monera, Protista, fungi, plant and Animalia. There is a comparison. You should read this lesson in a comparison manner by drawing a chart like this. It may seem little messy, but you should read like this itself. So you can formulate your own box or differences based on your understanding. You can use these notes as a reference. but you must read these kingdoms by using only boxes like this. So Monera cell type was prokaryotes. Protista is eukaryote. Unicellular eukaryotes comes under Protista. Next one is fungi is also eukaryotic. Fungi is also eukaryotic. And Planta is also eukaryotic, and Animalia. You should read like this. And cell wall Monera is non-cellulosic. And here in fungi it's non-chitinous. Chitin, it contains chitin. So, but it's non-cellulosic and present. Whereas in plants, there are cellulosic cell walls will present, and in animals it's completely opposite. So reserve food is important here. You should remember this. Next, nuclear membrane is also important. An organization, okay. Then mode of nutrition. Here, Monera, you can see autotrophic organisms and heterotrophic organisms. In autotrophic also, all types of autotrophic, that means complete all the different types of nutrition is seen in Monera. They may ask this question. All type of nutrition are seen in which kingdom? That is Monera. So next you have to remember remaining mode of nutrition. And I wrote subdivisions here itself. In Monera is divided into archaebacteria. Archaebacteria. Cell wall contains pseudomurine. They have branched chain lipids which help in survival of extreme conditions like halophytes. Thermoacidophiles, methanogens helps in biogas formation present in cow stomach. That means methanogens comes under archaebacteria. Their cell wall contain pseudomurine. Next, eubacteria cell wall contain murine. Eubacteria cell wall contain murine. Next, mycoplasmas. Mycoplasmas lack cell wall, and they are very small organisms. They have other name called PPLO, pleuropneumonia like organisms. And fourth point was actinomycetes. Their cell wall contain mycolic acids. So pseudomurine present in archaebacteria, murine present in eubacteria, and mycolic acid present in actinomycetes. And there is important point called radial colonies in culture. Which of the following subgroup forms radial colonies in culture? They may ask like this. Then you have to answer actinomycetes. Next, the genus Streptomycetes produces antibiotics. Most of the antibiotics are produced from the genus called Streptomyces. And there is other important, most expected bit was red color of Red Sea is due to Trichoderma erythraeum. Actually, blue-green algae is not an algae. It's a bacteria. It's a cyanobacteria. For example, Nostoc and Enabiana. And some important point about this Enabiana was during asexual reproduction, harmogonia, that is also called echinates, or fragments are produced. It contains central centroplasm. And peripheral chromoplasm. It contains central centroplasm and peripheral chromoplasm. Peripherally, they contain colored pigments. That is why it is called chromoplasm, pigmented. Centrally, it is colorless. Peripherally, it is colored. So the second one, Protista has some divisions called chrysophytes. dinoflagellates, euglenites, slime molds, and protozoans. Here I enlisted the important points of these subgroups. Chrysophytes include diatoms, desmids, which is also called golden algae. So they form diatomaceous earth. Diatomaceous earth is formed after death of these diatoms. It's also called Kishalgar over years. So there are two symmetry seen in these diatoms. One is central diatoms and pineal diatoms. Central diatom shows radial symmetry, pineal diatom show bilateral symmetry. Dinoflagellates, red tides in the Mediterranean Sea is due to dinoflagellates. Goniolax, Noctiluca, Mesocaryon. Mesocaryon is very important. Mesocaryon is a condition, condensed chromosome. They don't have H1 protein even in interphase. Generally, H1 protein helps to organize the chromosome, but here H1 protein is absent, therefore the chromosome was condensed. That condition is called mesocaryon. Euglenoids, instead of cell wall, they have protein rich layer is called pellicle. Okay, it contains a protein rich layer called pellicle. They are called actually mixotrophs because they perform both photosynthesis. They are photosynthetic and heterotrophic also. They can prepare their own food and depend on others for food. So the fourth group was slime molds. In slime molds, in suitable conditions, they form plasmodium. In adverse conditions, spores with true cell walls. Protozoans are relative to humans and they lack cell wall. Example, Trypanosoma, Plasmodium, Paramecium. These are very important. Ciliated protozoans was Paramecium, flagellated protozoans was Trypanosoma, and sporozoans are Plasmodium. Fungi has some other groups. Mainly this is important. Fungi has some association with algae that is called lichens. And fungal part of that lichen is called mycobiont. And algal part of that lichen is called phycobiont. So fungi plus roots of higher plants. Fungi can also associate with the roots of higher plants. That is called mycorrhiza. Next, asexual spores are conidia, sporangiospores and zoospores. How can you remember this? Asexual spores A to Z SC. You just remember A to Z SC. It means asexual spores, A for asexual spores, Z for zoospores, SC sporangiospores and conidiospores. Sexual spores are, for sexual spores you can just remember like this, AB. That means O for oospores, A for ascospores, B for basilospores. This is sex spores. So they may ask you which one of the following is the example for sex spores. They will give all these options. Then you have to remember that the example for sex spores was this. Then you can simply say oospores, ascospores and basilospores are the examples for sexual spores. For asexual spores, the hint was A to Z SC. A for asexual spores, Z for zoospores, S for sporangiospores, C for conidiospores. Next, the four other examples outside subgroups of fungi was Phycomycetes, Anascomycetes, Basidiomycetes, and Deuteromycetes. Phycomycetes has other name called algal fungi. Ascomycetes has other name called sac fungi. Basidiomycetes is also called club fungi. And Deuteromycetes is also called imperfect fungi. So Phycomycetes has some other examples: Rhizophos, Albugo, Mucor, Pinmowl. You have to remember all this. It's very important. Examples plays a crucial role in this lesson. So in plantae includes algae, bryophytes, pseudophytes, gymnosperms and angiosperms. And Animalia is more advanced group. So here Phycomycetes, Ascomycetes and Basidiomycetes has some other additional information. So I wrote that here. Phycomycetes are coinocytic. Coinocytic means that hyphae has no septum and they contain many nucleus in it. So that is called coinocytic. Aseptate. There is no septum. Septum means separation. Like this. Mycelium, as we know. So next, spore formation can be sexual or asexual. Both are endogenous. You should remember this point. Sexual and asexual spores are endogenous. But in Ascomycetes, mycelium was branched and septate. That means it can show branches and septum also. Spores are sexual and asexual. Asexual spores are exo. Sexual spores are endo. Next, they show some fruiting bodies which are called Cleistothecium. Cleistothecium means globe, no opening, they don't have any openings. Perithecium, flask shaped, they show flask shaped structures. And apothecium means they show cup like structures. You should remember this. Global for Cleistothecium, perithecium is for flask, apothecium is for cup. And Basidiomycetes are mycelium is branched and septate. Spores are sexual and asexual. Sexual are exospores. Asexual spores are absent. You should remember this. Asexual spores are absent or sexual spores are endo, but here it's exo. It's a hint. All sexual spores are actually endo, but here sexual spores are exo in Basidiopspores. So in Deuteromycetes it's opposite. Sexual spores are absent and sexual spores are exo. Exactly, it's opposite to Basidiomycetes. So this was very, very important. And here it is about euglenoids. Euglenoids, we already know that it contain eye spot and it contain invagination, that contain cystosome, cytopharynx and reserve. It contains an eye spot that is photosensitive stigma. That is also called photosensitive stigma. That means it is sensitive to light. And it contains a protein-rich layer called pellicle, which helps in flexibility. See, euglenoids grouped under Protista. This line was very important. That can clear much confusion. Grouped under Protista. Actually, euglenoids are grouped under Protista in this group, but might tell you it is an algae. Boundaries of Protista is not well defined. It is a Protista, but some say it as algae. It is actually algae. Euglenoids is actually a Protista, but some may include euglenoids as an algae. So here these three are very important. Scrape disease of sheep is caused by prions, and potato spindle tuber disease was caused by viroids, and mad cow disease is caused by prions. From prions we can see scrape and mad cow. Scrape of sheep, mad cow disease can also seen for because of prions. Potato spindle tube for viroids. So chapter 2 biological classification was almost done. This point was very important. I will end this class with this important point. So for fungi, PABD, that means please accelerate better decomposition. It's a hint. Please accelerate better decomposition. Please for Phycomycetes, accelerate for Ascomycetes, better for Basidiomycetes, D for Deuteromycetes. Here you write sexual and asexual. In asexual Ascomycetes, you can write ascospores and basidiospores. For Basidiomycetes, it's basidiospores. In asexual, Phycomycetes shows sporangiospores, which can be zoospores or eplanospores. In Ascomycetes, asexual was conidiospores. In Basidiomycetes, there was no asexual reproduction. In Deuteromycetes, they show conidiospores. Asexual spores for here I gave some Z, A to Z, SC. A to Z, SC. A for asexual, Z for zoospores, S for sporangiospores, C for conidiospores. These are the questions. Try to solve these questions. This was a simplified one. Exogenously produced conidiospores and basidiospores. They might ask you which of the following was exogenously produced. That was conidiospores and basidiospores. Which was endogenously produced? Rest of all. Rest of all are endogenously produced. That's it for chapter 2. You can use this table as a reference, but you should read this lesson in this way itself. Otherwise, you can't. It will feel difficult to remember this chapter. So form a table and write the details according to you. It can be seen little messy, but you can formulate your own notes by using this as a reference. Okay, thank you. Let's meet in the next class.